Protein synthesis consists of two processes, which are transcription and translation. The first is transcription. Transcription is the process of copying DNA information into mRNA. This process occurs in the nucleus of eukaryotic cells. The first stage of transcription is initiation. In this stage, the enzyme RNA polymerase binds to the promoter sequence upstream of the DNA. The upstream region is called the promoter, and it is high in adenine and thymine bases. The DNA strand is unwound, and hydrogen bonds are separated, exposing the template strand. The second stage of transcription is elongation. RNA polymerase binds mRNA 5' to 3' on the new mRNA strand after the promoter sequence by adding complementary nucleotides. Unlike DNA replication, RNA does not require a primer in this process. The RNA also does not require both strands of DNA. The coding strand remains unused because it matches the RNA strand being made with uracil in place of thymine. The template strand is used as a template for RNA polymerase. The final stage of transcription is termination. RNA polymerase continues to build mRNA until the termination sequence is detected. At this point, RNA polymerase and mRNA are released from the DNA template strand. RNA polymerase becomes free to bind with another promoter region and transcribe another gene. After the 3 steps of transcription are complete, post-transcriptional modifications are made to the primary transcript in eukaryotic cells. The 3 modifications are 5' capping, poly-A tail addition, and splicing. A 5' cap is added to the start of the primary transcript, replacing the triphosphate group. This prevents the mRNA from being digested and allows binding to the ribosome during translation. A poly-A tail, which consists of a string of about 200 adenine ribonucleotides, is added to the 3' end by a poly-A polymerase. This again prevents digestion and increases stability of the molecule. Splicing is when introns are removed from the primary transcript. Introns are non-coding regions and are spread out amongst the exons, which are coding regions found in the mRNA. Spliceosomes remove the introns before translation to avoid ineffective protein folding. After all of these steps and modifications, the mRNA is finally ready to leave the nucleus and is called the mRNA transcript. The steps of translation will be explained next.
Now that transcription is complete, translation will now turn this mRNA transcript into a protein. Once the mRNA has left the nucleus, it enters the cytoplasm with its new cap and tail attached at the ends. Once in the cytoplasm, the mRNA searches to find the organelle that is in charge of protein synthesis. This would be the ribosomes. As the mRNA reaches its destination, 2 ribosome subunits bind to the 5' cap end of the mRNA, having the big subunit on top and the small subunit on the bottom. Then initiation begins. Initiation is the start of translation by the ribosome subunits attaching to the mRNA and being located in the cytoplasm. The ribosomes begin to slide across the mRNA using the 2 sites on the ribosomes for tRNA to attach to. tRNA is called transfer RNA and is the link or adapter to the mRNA and helps build the chain of amino acids to make up a protein. These 2 sites are the A site and the P site. Translation commences once a start codon reads the code AUG. AUG is the start codon for translation in every single cell. The second step of translation is elongation. When the start codon is in the P site, a tRNA delivers the amino acid methionine and recognizes the code because of its complementary anticodons. A second codon attaches at the A site and another tRNA delivers the next amino acid in the protein sequence. Once the 2 codons are attached to the A site and the P site, a peptide bond is formed between them. As the peptide bond is formed, methionine is shifted to the E site, which is our last site on the ribosome. The E site releases the start codon in order to obtain another amino acid to build the chain. The second codon is now shifted to the P site, and a new codon attaches to the A site and waits for an amino acid to bind and add to the protein chain. This process continues across the mRNA transcript until it reaches a stop codon. This stop codon would be a sign of termination, which is our last stage of translation. A stop codon is 3 different triplets that stall the ribosomes and stop the growing polypeptide. When the stop codon is attached to the A site, it is a release factor because it does not have an amino acid attached to it. The codon forces the large and small ribosome subunits to disassemble from the mRNA in newly formed protein. Translation is now complete as we have created a polypeptide chain for our mRNA transcript. This protein starts the process of protein folding and finds its way to the Golgi complex to be stored for the body. In conclusion, that was the complex process of transcription and translation, and we hope you understand how DNA information can transform into mRNA and then a protein chain. We hope you enjoyed.